Good morning everyone. I'm recording this video in Riga, Latvia, as I will be live from Stanford, San Francisco on 16th of May. At this moment, I will be visiting Stanford for a few days, but the pictures that you see on the screen is my new workplace in Riga. Today I'm going to give you an update on our ongoing work where we are comparing the biological features of both ME/CFS and Long COVID. Understanding the initial mechanism of disease development is key to the treatment strategy. We all went through the pandemic and now we are all very familiar with the term Long COVID. Sadly, people are now starting to recognize ME/CFS as they find very similar clinical features in the old lost twin brother ME/CFS. But are these two conditions exactly the same? The answer would be yes or no. The reason lies in the origin of the disease. For Long COVID, we all know the virus SARS-CoV-2. And majority of the Long COVID patients are still within the first few years of the disease. But unfortunately, the story is not the same for ME/CFS. The disease diagnosis is often very late for ME/CFS, and by the time the disease is diagnosed, the pathogens are mostly undetectable in the body fluid by any of the commercially available techniques. What remains is the chronic manifestation of the clinical conditions. I'm sure over the next 3 to 4 years, many of the Long COVID patients will become ME/CFS without even being officially declared as a Long COVID patients. Today I'm not going to talk about herpes viruses or mitochondria, which I often talk about. I'm going to talk about two important features of our body. First is natural antibodies, particularly the natural IgMs. These antibodies are induced, or they are not induced by antigens. They are inherently synthesized in our body starting from the placental stage. A very specialized group of cells called B1A cells produce these antibodies in primary hematopoietic tissues. They are often structurally different than antigen-induced IgMs as they exist in the form of pentameric molecules. Their job is to clean the body from pathogens and cellular debris. Therefore, they prevent the development of autoimmunity. I will show you today how these natural IgMs are depleted from the blood circulation after COVID-19 infection, and how this is a key difference when we compare Long COVID to ME/CFS. I will focus on a very specific natural IgM which is developed against fibronectin, and we call it as a IgM FN1. There are only a handful of papers available in the literature that talks about this particular natural IgM against fibronectin. Here is one old paper where the authors show that trypanosoma infection, which is a parasitic infection in mice, depletes the natural IgM against fibronectin. And when the mice recover from the infection, these natural IgMs are also recovered. As this particular natural IgM recognizes fibronectin, it has an inverse correlation with fibronectin amounts. That means when IgM against fibronectin is low, just after the infection, the fibronectin is high. And after recovery, when IgM against the fibronectin becomes normal, FN1 levels also come down. Interestingly, fibronectin protein itself binds to many viruses like HIV, or bacteria like Borrelia or Staph aureus. So what is fibronectin? Fibronectin exists in three major forms. The most common is plasma fibronectin. It is a soluble fibronectin produced only by hepatocytes, and it contributes to blood clotting. The second type is the cellular fibronectin, which is one of the most important components of the extracellular matrix, or ECM. This is produced by many different cell types and plays a key role in tissue healing. The third type of fibronectin is the alternately spliced variants of the fibronectin, which contribute to tissue remodeling, fibroblast differentiation, inflammation, and also even in tumor progression. Even though fibronectin is so important for our body, too much of it also is not good. A fine balance between fibronectin amount is necessary. It has been shown that high fibronectin is often found in Alzheimer's patients, and it accumulates on the blood vessel and blood-brain barrier, causing damage to the blood-brain barrier. Today we are going to focus on the relative values of natural IgM against fibronectin and the total amount of fibronectin in the serum. We will talk about the ratio between these two values and how it correlates with long COVID disease severity. Just to keep in mind that these natural antibodies should be there in the body under normal circumstances, and it gets depleted after COVID infection. The third type of fibronectin is the alternately spliced variants of the fibronectin, which contribute to tissue remodeling, fibroblast differentiation, inflammation, and also even in tumor progression. Even though fibronectin is so important for our body, too much of it also is not good. A fine balance between fibronectin amount is necessary. It has been shown that high fibronectin is often found in Alzheimer's patients, and it accumulates on the blood vessel and blood-brain barrier, causing damage to the blood-brain barrier. Today we are going to focus on the relative values of natural IgM against fibronectin and the total amount of fibronectin in the serum. We will talk about the ratio between these two values and how it correlates with long COVID disease severity. Just to keep in mind that these natural antibodies should be there in the body under normal circumstances, and it gets depleted after COVID infection, and the fibronectin amounts should be balanced or low in the body, but when they go up, they are associated with the disease. This slide may be familiar to some of you, as it shows the first cohort of samples we started working with. It is already available as a preprint in medRxiv. Our initial study group was healthy controls, ME/CFS patients, which also includes COVID-induced ME/CFS patients and sclerosis patients as a disease control. We compared this to mild, moderate, and severe long COVID patients from our Würzburg COVID cohort, which is a part of the National Pandemic Cohort Network, or NAPCON, from Germany. The long COVID severity scale is based on a published scoring system from our German colleagues, which was later also published by the USA Recover study. We submitted this paper last July, and it was reviewed by a top journal, and the reviewers had a major suggestion for us. They asked about validating our results in independent validation cohorts. This was an encouraging idea, but was way too challenging than we thought. It took us almost 7 to 8 months just to go through the entire world to find the best biological samples from clinics following similar selection criteria that we were using, and going through ethical permission, getting the samples transported to Germany, and all this without a single penny of funding from anywhere. But we did it. For ME/CFS, we found 2 validation cohorts. One is from the Riga clinic, Sweden. This sample cohort was small but very encouraging for us, as these were freshly collected samples, only 18 to 20 months being stored in the freezers. The other cohort of ME/CFS patient was from UK ME/CFS Biobank, and we are very thankful to both these sources to provide us the validation cohort. For the long COVID validation, we found a larger cohort within our German NAPCON biobank. This cohort was from Kiel in Germany. We could have used hundreds or even thousands of samples from these NAPCON cohort, but as I mentioned, we are restricted by funds, and the time to do these studies are also very limited. So we focused on 300 additional long COVID samples from Kiel cohort. Before we go details into the results, just one slide of the result I will share with you. But even before that, I would like to thank especially 3 of my colleagues. Stefan from my lab, he is a PhD student within my group, who has done all these experimental studies. Sharda from Stanford and Olga from University of Würzburg, who are doing all these data analysis and modeling and machine learning, etc., and making it possible to present these results to all of you. Now here is a slide that shows a few cow plots. You can see that it is actually possible to separate long COVID or ME/CFS from healthy controls, and even it is possible to separate them from other disease controls. I'm showing here only the fibronectin amounts in the serum, because this is the most prominent value that can differentiate in both long COVID and ME/CFS. The left panel here shows the fibronectin values in the total long COVID patients, including the test cohort as well as the validation cohort. And you can see that healthy controls and severe LC patients are nicely separable from each other. And even though mild and moderate long COVID groups overlap with the severe long COVID groups, they still occupy a middle position in between the healthy and the severe long COVID patients. For the ME/CFS cohorts, you can see that fibronectin values can separate the ME/CFS patient from the healthy controls and even from MS patients, which lies somewhere between the 2 groups. The separation is not very prominent in the UK cohort of samples, as in Germany and Sweden ME/CFS cohorts. Now let's focus on individual cohorts. Here are the kernel density plots showing fibronectin amounts on the top and the IgM fibronectin amounts on the side, the y-axis. These 2 cohorts are from Germany. On the left side you have the Würzburg cohort, and the right side is the Kiel cohort. In both the cohorts, you can see that the serum fibronectin amounts are high in long COVID patients. The Kiel cohort, which is the largest cohort, particularly shows a clear increase in serum fibronectin amounts in long COVID patients. The severe long COVID patients, here in the green line, show slightly higher fibronectin amounts in comparison to the mild and the moderate long COVID patients. As you can see, when fibronectin is getting increased in long COVID patients, the natural IgM against fibronectin decreases in these patients. So these are log2 fold values being shown. If you want to look into this overall distribution of the ratio between these 2 values, the fibronectin to the IgM fibronectin values, you can see in these kernel density plot, the healthy controls, which is shown in the red over here, distinctly occupy a different area than all the long COVID patients. The ratio between these 2 values is shown in this cow plot over here, and you can see that the healthy controls stand complete separately from all the long COVID groups. One thing to realize here that there are no major differences within the different severity of long COVID groups. It seems that these values are changed in response to the SARS-CoV-2 infection and not to the extent of the clinical symptoms, the severity of the disease. We also have to remember that these values are within the first 6 to 12 months after the COVID infection. Now let's have a look on the same FN1 to IgM FN1 ratio in the ME/CFS patients. As mentioned earlier, fibronectin values have a clear trend in ME/CFS patients in all the 3 cohorts. They are increased in comparison to the healthy controls. But except for the Swedish ME patients, we do not see much difference in the IgM values against fibronectin between healthy and ME/CFS patients. This is very much dependent on the actual onset of the infectious origin of the disease, as IgM values recover over a period of months to years after the first infection. I mentioned that the Sweden cohort of samples was comparatively fresher than the other 2 cohorts being collected within the first 18 months from the date of laboratory analysis. That might also be one of the important factors to see these differences only in the Sweden cohort, not in the Würzburg and the UK cohort. Of course, we see a trend in UK cohort, but not exactly clear differences. We had the opportunity to follow a small cohort of long COVID patients from Würzburg for 24 months after the first COVID-19 infection. Their baseline visit was within 6 to 12 months after the first infection. He is presented as C1. And their follow-up visit was after 12 months of the baseline visit, which is basically around 24 months after the first COVID infection. And we measured both fibronectin and IgM fibronectin levels in these patients, and you can see the ratio between IgM fibronectin remains more or less the same in mild long COVID patients. So this is the visit 1 and the visit 2. In the red you can see the mild long COVID patients. They remains more or less same. However, in moderate and severe long COVID patients, this ratio further goes down. Olga and Sarda tried to develop a transition model to interpret these values. Let's divide the entire range of IgM FN1 to FN1 ratio into 4 quartiles. So Q1, Q2, Q3 and Q4. Q4 is the highest ratio, means better, while Q1 is the lowest ratio, means it's worse. You can see that majority of the mild long COVID patients, which is presented as an orange box over here, they lie in the quartile 4 during the first 12 months time. Over the 12 months follow-up time, these quartile 4 mild patients improved and their ratio still remains in quartile 4 or decreased. Only a small number of these quartile 4 mild patients actually their values worsened and went to the quartile 2. For those mild patients, very few mild patients having worse ratio or bad ratio in the first baseline visit, they improved and they came back to the quartile 4 after 24 months. You can see that the quartile 1 patients mostly remains mild, the moderate and severe patients, they remains in quartile 1. The quartile 2 patients mostly worsen and fall into either quartile 1 or quartile 2. Some are into quartile 3 also, and quartile 3 patients, they either remain in quartile 3 or worsen to become quartile 2 or quartile 1. So after 24 months, you see the number of patients in the quartile 1 and quartile 2, which is the worst state, increased. So long story short, the ratio between IgM FN1 and FN1 perfectly correlates with the PCS score-based differentiation of the long COVID patients, which is published by our colleagues in the past. Mild long COVID patients, they recover the natural IgM levels and fibronectin levels come down over the 24 months time. However, moderate and severe long COVID patients, particularly the severe patients, continue to drop their natural IgM levels and the fibronectin amounts keep increasing, leading to a poor ratio between these two, which decreases over 24 months time. We are still waiting to know how these patients were in terms of their disease severity in 24 months follow-up visit, so that we can see if this ratio that we are talking about can actually predict the disease or not. I would like to acknowledge the help from many of my collaborators. Without those help, we would not have managed to do these studies. Carmen and Franziska from Charité Berlin helped with ME/CFS samples from Berlin. Gabby provided the sclerosis samples from Lübeck. Per and Dawn from Braga Clinic, they provided the ME/CFS cohort samples from Sweden. And then we have Louise, Eliana, and Caroline from UK ME/CFS Biobank. They provided the samples from UK. Niklas and Caroline Morbach from our Würzburg clinic, they helped me out with all those ethical approvals for long COVID studies from Würzburg. Thomas Bahammer from Kiel is crucial for our success to get the samples from Kiel. And I would like to mention Peter Heusmann and Olga particularly, and they're instrumental in the data analysis from the Würzburg. And Sarda played a key role from Stanford doing this mathematical modeling and things like that. This is my small team who put together all this work, even though we were never been funded for LC studies, long COVID studies. However, my greatest thank goes to Binod Khosla and Amar Foundation for the career development grant they provided to me, which is the lifeline for all this work which is going on in our lab. I am also thankful to BMBF from Germany to fund the ME/CFS studies within Germany and all the long COVID samples from the NAPCON cohort. Last but not the least, thank you, all of you, for your support and most of all, being there with your kind words when times were not very good and demoralizing for us. So thank you for your attention, and I hope that I will be available online at this time to answer your questions. Thank you.
Thank you very much, Professor Prusty, for this glimpse into your research, but also for a glimpse into the conditions under which you conducted this research. It kind of became clear the severe lack of funding, and this is something that definitely has to change. We need more funding. I'm grateful to you for doing this research nevertheless, but this is something that has to change. Thank you also for being here now to answer questions. And I'll start right away because there are a couple. The first question concerns IVIG. Do you assume that IVIG might help with IgM depletion?
Yeah, personally, from scientific point of view, if I understand the science properly and if I'm correct, yes, it should help those patients where the natural IgMs are depleted, but it all depends on the IVIG composition, how it is prepared and how it is being given. From clinicians who have success in IVIG treatments, I have seen that there is some sort of a side effect in some of the patients. This is just because how you give the IVIG to the patients, and it is not on the basis of some side effects to the treatment itself. So, yeah.
Mm-hmm. Okay, thank you. The next question is very specific. Regarding long COVID, is there a difference in symptoms between patient cohort of those who caught COVID infection pre-Omicron versus during and post-Omicron?
Good question. As I mentioned that we had to focus our study on very critical questions depending on what funds available and how we can finish sooner. So we focused on pre-Omicron variants. The problem with the post-Omicron infected patient is that at that time we had vaccines and we had multiple rounds of infection. It was almost difficult to characterize these patients clearly, what is going on, what other type of exposures these patients had. So we try to avoid these patients at this stage, but I fully agree with you that if time permits and if funds available, we would love to test these patients after the first round of COVID infection. Yeah.
Thank you. Next question is from a patient who was diagnosed with ME post-COVID vaccination. And she asks, do you think the mechanism of action is the same for long COVID and for vaccine-induced long COVID, of which are leading to an ME CFS diagnosis?
From the clinical point of view, we have heard many talks, and it seems that there is a lot of overlapping between in clinical conditions among these patients. But the underlying mechanism, which is of interest for me, probably is the same. It is all about the inflammation, is all about the cell danger response, all about the dysfunctional mitochondria and things like that. And in most cases, it is all combining at the end to the herpes virus reactivation also, which we did not talk today. But it seems the root cause or the mechanism of development of these clinical conditions at some point overlap with each other. Yes.
Thank you. Next question. Do you believe that long COVID and ME patients have multiple simultaneously active infections?
This is a hard question to answer. Yes, there are many literature, many authors that try to believe or their own opinion on different type of infections. But what I believe is that if we cannot prove the mechanism by which an infection can cause disease, then we should not dilute our idea about the infection, because it can be cause and can be effect. So we focus on certain viruses, certain mechanisms where we can prove the consequence of the infection on the disease development. And that's how we can really validate the infection as a cause of the disease. Yeah.
Thank you. The next is a very overarching and important question. The person asks, most presenters have shown differences between ME and healthy controls, albeit with many different types of tests. Now, how can all this information be collated together to inform a united hypothesis and therefore targeted treatments? It seems that we have enough data to describe the disease already.
It's true. There is enough data, and there are many good minds around the world working on solving these issues. The problem is that these are one individual pieces of stone lying on the side of the river. When they combine together, they come to make a bridge. They can stop the flow of the river. So it's just required to bring them together and put forth as a combined effort and combined view, and then only people will start hearing about it. Yeah.
And do you have a suggestion on how to bring it together to form the bridge?
This is one. Today we did it in last 2 days. This is one good effort. But I think things are changing slowly, and I'm pretty confident that pretty soon the whole flow of water is so the ideas are so united now, as we heard in last 2 days. There's no way one can say that they are not making any sense. Yeah.
Thank you very much. Time is up. So thank you for being here.
Yeah. Thank you for having me here. Yeah.